Good morning, class. Welcome to the unit on research methods. We're going to do this in two recordings, with the second part primarily focusing on brain imaging methods. For this first part, we're going to get into a general overview of animal methods in neuroscience, in addition to some just general overview of the scientific process. So research relies on empirical data as its means of acquiring knowledge. This would involve, typically more than, just interoceptive thinking or just writing down one's thoughts. This would typically rely on the scientific method, which involves the hypothesis testing of theories. It involves operational definitions. For this, it's important to note, what is the operational definition of a term? There are so many good examples out there, but for example, one could say, we're going to go out and study whether or not it's hot outside. So, we would need an operational definition of what it means to be hot outside. One person could say, that is, 80 degrees, another 70 degrees, another 100 degrees. So understanding what the definition is fundamentally important. And you see this a lot, I would say, really, even amongst experts. They tend to be arguing over different definitions, thinking they're talking about the same thing. You certainly see it all the time on the news. I would say it's one of the most common things that you're going to find is that people are really not arguing about what they think they're arguing about. They have different names for the same thing, and are really arguing different points because they have different definitions. This is also, an opposite approach to relying on common sense or folklore, which, may or may not be true. Science also differs from folklore and tradition because it uses empirical methods. You would seek to quantify something rather than, hedging your bets on, some superstitious, car approach. Or, here's a good one. I remember being taught back in the day that, you should pour a Coca-Cola on your car battery, when it has a little corrosion built up on it. So, there is some logic in there, and that, some acid, could possibly get rid of other acid in some, but yeah, for the most part, that doesn't work. All that results in is you having a sticky battery that also has corrosion on it. That would be an example of a sort of a tradition that was passed down that wasn't a very logical tradition, even though it was based somewhat on some reasonable evidence. So science, and thus neuroscience, is tentative, meaning that it's based on current information. This is always going to need to be updated. I was taught when I was your age that there's only one neurotransmitter per neuron. We now know that this is very much incorrect, and there are numerous chemical signaling molecules within one neuron. There are countless things that are being updated in neuroscience, and the field is always evolving. So you must stay abreast of new changes in the field, and that being said, You know, I think oftentimes students expect there to be more turnover than there is. You know, it's also true that science is quite slow. You know, we might assign an article from 15 years ago, and you would think that it's been completely, you know, outdated by then. In reality, it's just barely been replicated. Oftentimes, you know, we still don't even know if this is, you know, replicable yet, let alone something we should throw out. So, you know, you do have to have some expectation of the slowness of all of this. You know, that being said, neuroscience wasn't even a field 100 years ago, and it has grown incredibly rapidly since then. There are many rules for conducting scientific research. The ethical ones are growing. You know, one of the big ones involves animal research. You know, some of the animal research that I've done will probably not be legal within 50 years or so. You know, I would say that's already the case in some countries in Europe. There are many other ethical considerations out there to research involving human research. You know, it used to be more common to try experimental approaches and treatments and ideas out on prisoners, on enlisted military people, you know, on the poor. So now, you know, a lot of those things still do occur. It is worth noting that it just looks different. Um, you know, we still have to try out new drugs on people before they are passed. So instead of doing those in prisons in the United States, we now outsource this to poorer countries. So I mean, like, and then we don't look too closely into their rules of, you know, who is signed up for these studies. And there are lots of things that you know, we've done in the past in this country that are still accepted elsewhere that I guess we'll sort of turn the blind eye to if we outsource the clinical trial to that country. So you know, I think that many of the things that we are doing now, you know, will be considered unethical to your children, just like many of the things that were done before us now look more unethical now through modern sensibilities. We're going to go over a variety of scientific techniques, so you have some sort of assessment or understanding, appreciation for what's out there. You know, and then we're here right now. I'm going to briefly talk about two main methods of experimental research. One being primarily correlational in nature, the other being some application of the experimental method. Here, I'm saying that we would have a hypothesis, some sort of independent variable that we would manipulate and then observe a dependent measure. So here's a example of your correlational methods. This would be non-experimental. Here you'd be looking at a relationship between, you know, two variables. You know, is the SAT predictive of the GPA? So I mean, like, you know, if you look at SAT on one side and then college GPA on the other axis, that would be giving you correlational information. This does not imply causation, so that would not mean that your SAT score is causing you to have a good GPA score. Just that you know, these two oftentimes go hand in hand. The closer that you get to one in terms of your correlation, so typically when you have your axes and then a whole bunch of data points in between them, and then you draw a line. So you know, a strong correlation would appear closer to one than to zero, or to -1, which would be an inverse correlation. So positive correlation goes from 0 to 1. As x goes up, so does y. For example, the rate of water skiing might increase, as does the temperature. A negative correlation would go 0 to -1. As x goes up, y goes down. So the rate of hot chocolate intake might go up as the temperature outside goes down. You know, here are just some representative correlations, college GPA and SAT score, hours slept through age in years, and number of houseplants by the number of times brushing teeth. So there you would see no correlation, you know, up to the far left, a positive correlation, and then to the top right, a negative correlation. Now let's talk a little bit about the opposite, experimental methods. You know, this can allow us to conclude causation. Having done this myself, typically we are still just talking out our asses. We typically don't actually have causation, but you know, we're trying to get toward that, and at some point we also put on the sales pitch, you know, part of, you know, dissemination of anything, whether it be a new invention or scientific data. You know, there is showmanship to it. You know, you want to, you know, and what's more impressive than showing that you can show causation. You know, we typically say that we do this with optogenetics, for example, a technique that I'll tell you about in a little bit, where you can drive neurons and then see a behavior manifest. But still, I would argue that that's not necessarily causal. You know, you're seeing a correlation between the firing of that neural population and a behavior. You don't necessarily know 100% that the neurons are driving that particular behavior. First of all, that behavior could be a compensatory behavior for some other physiological or behavioral mechanism that you're primarily driving with that neural sequence of events. Just as one example. But anyway, you know, experimental methods generally use a hypothesis and then use manipulation of an independent variable to observe changes in a dependent variable. So you need independent and dependent variables in an experimental design. A variable would be any characteristic or condition which is subject to change. An independent variable is what the experimenter manipulates to change, and then the dependent variable is what the experimenter measures, what was changed by the independent variable. You know, so for example, right, the independent variable might be, you know, how many tests I give. So I might propose that if I give you 10 exams rather than 2 exams, you will spend more time studying this year. Well, you know, somewhat of a flawed experiment, depending on the number of tests. You know, we could certainly manipulate these variables, and I could increase the frequency of tests delivered, and then monitor how many hours studying you spend. And then we can either support or reject our hypothesis that more exams equals more studying. These often involve control groups that do not receive the experimental treatment. You know, so for, right, there are a variety of potential control groups, especially when we think of pharmacology and drug experiments. We tend to have a placebo group that thinks they got the same treatment. This is a little difficult when it comes to psychoactive drugs. You know, you're seeing a lot of information, a lot of news, excitement these days about psilocybin studies involving magic mushrooms. I think that these in particular are difficult because even those that say they're placebo controlled, you know, it is difficult to, you know, mask whether or not you are having visual hallucinations. You know, I guess if you are dealing with, you know, subthreshold doses or doses that are not producing massive hallucinations, and maybe, but you know, it becomes pretty hard to control, you know, create placebo-controlled trials for some of the drugs that we study. Still, the placebo-controlled trials are, very important. And placebo effects are very high in psychiatric medicine. There have been some recent studies to demonstrate that the antidepressant effects of serotonin, selective serotonin reuptake inhibitors, could be as high as 50%. So, in about 50% of patients taking SSRI and thinking that it is treating their depression, that's probably only true in about half of those patients. The half are thinking that they are improving because they're on any sort of sugar pill. So, what are correlational versus causal relationships? In correlational, you would have there'd be no random assignment to groups. You're just looking at two things. Does SAT predict GPA? Does gender predict GPA? Are men better than women at this? Are women better than men at this? These would be simple correlations that don't really, mean too much other than giving you sort of an understanding of the groupings that we're in. So in causal relationships, you would tend to have some sort of random assignment into a placebo group or a control group or like the actual experimental group itself. And then a researcher would manipulate the independent variable. In the field of pharmacology, typically you would want at least 3 doses. So you would have the potential to see an ascending or descending trend. And you would actually know where you are in terms of that trend at any specific, measurement. And these sorts of placebo control groups are important. Well, this isn't a placebo example, but a good control example provided by Craig Bennett. Dr. Bennett purchased a whole Atlantic salmon and decided to put this into an fMRI machine that was used to study the brain. It was to be the lab's test object as they worked out some new methods. However, as the fish sat in the scanner, they showed it a series of photographs depicting human individuals in social situations. To maintain the rigor of the protocol, and just like the human test subject, the salmon was asked to determine what emotion the individual in the photo must have been experiencing. The salmon, importantly, was also not alive during the scanning session. But when analyzed, the voxel, so really these are 3D pixels, fMRI images, it's very much like looking at these sort of like pixel art programs. They sort of just go in and emphasize and analyze the amount of voxel pixels being expressed in a specific region. But anyway, the voxels representing the area where the salmon's tiny brain sat showed evidence of activity. It looked like the dead salmon was actually thinking about the pictures it had been shown. By complete random chance, they found that some voxels that were significant just happened to be in the fish's brain. And, if I were a ridiculous researcher, I'd say a dead salmon perceiving humans can tell their emotional state because, such things could be concluded without controls. So, this result is completely nuts, but that's actually the point. Bennett and Wolford wrote up the work as a warning about the dangers of false positives in fMRI data. They wanted to call attention to the ways the field could improve its statistical methods, and researchers get up to 130,000 voxels from each set of scans. They must comb through all those data for signals that indicate something is happening in a particular region of time. The fMRI data has a lot of natural noise, and if you really wanted to find some results in that mess, go on a fishing expedition. You could probably find the fish you wanted to. And that would be, what's known as a false positive, or you seeing something that, and it's a very common thing that you can see that, especially when you want to see it. You are seeking out to confirm your hypothesis, which is one reason that in certain fields like psychology, people that train you are very, keen to stress the idea of the null hypothesis, that you're trying to really disprove something rather than prove something. But it all gets back to this confirmation bias. Now, there are many different techniques in science. We're going to go over a lot of them. Some that I'm going to talk about now include cell work and a little bit on animal studies. Histology is the main type of cellular work done in the field of neuroscience. Your book goes into a lot of different stains. I'm not going to test you on those. I will point out that Camillo Golgi, the guy that also named the Golgi apparatus, developed the first and most important stain in neuroscience called the Golgi stain. Interestingly, he used this to argue that the brain isn't different neurons; it's all just one network. But then another younger scientist by the name of Ramon y Cajal, used the same stain to observe different cells of the nervous system, which is the interpretation that we basically go with today. But we know that because of histology. So histology involves perfusion. First, you would remove the blood from the brain. We do this using, a sequence of steps. Generally, you just pump the fluid out. You can buy nice pumps that do this to mimic the heartbeat. If you do things just right, you can, actually have the animal's heart beat the blood out of the system itself, and then replace it with formalin, as the heart stops. Then you would fix the brain in a formalin solution and let it sit there for some period of time. Typically, you wait until it floats. Then you would slice the brain into thin sections using a microtome and stain the specific sections using some sort of stain, like the Nissl stain, the Golgi stain. And again, I'm not going to quiz you on the different types of stains. How a brain is processed during the histological process is listed here. You would perfuse the brain and animal to remove blood from the tissue. The brain is then fixed in a formalin solution to harden the tissue. You're halting autolysis here. Essentially, the cells, in your body are going to be decayed by microbial life. And by fixing it with formalin, you are basically creating a tissue structure that cannot be decayed by microbial life. It also is chock full of carcinogens and nasty things that will harm your cells, so you have to be a little careful of it. The brain is then sectioned with a microtome. This, essentially is a deli slicer. Stains are applied to the sections to reveal the internal details of the brain, and the sections are then looked at under a microscope. Here are various microtomes. Some look more like a deli slicer than others, but at this basic level, that's what they are. They're essentially fancy deli slicers. We can also measure brain activity. This has been most done most famously using EEG. This was developed by Hans Berger in 1929, who recorded from 2 electrodes on a scalp over an area of interest. This involves an electronic amplifier that detects combined electrical activity of all neurons between these 2 neural points, and then you can graph the activity. This provides relatively poor spatial resolution, so it's not exactly clear where you're recording from, and it also isn't exactly clear what this represents. We know that these waves seem to be important, but there does seem to be there is considerable debate as to what they mean. Are they reflective of sort of signal going into a specific brain region or leaving a specific brain region? These I think are very important questions that we still need to answer. But what can EEG be used for? It's useful for detecting changes in brain patterns or arousal. You can average several readings to obtain an evoked potential. And this can often be used for detecting epilepsy and other brain disorders, such as sleep disorders. This is very common in sleep labs. In fact, I'd be willing to bet that your smartphone probably has a setup where you can even give yourself an EEG. And now I'm going to talk a little bit about stereotactic techniques. Stereotactic techniques allow precise positioning in the brain of electrodes or other devices. Very similar apparatuses are used in people and in animals. This allows us to, put an implant or perform a measurement in a specific brain region. Um, one of the big differences that would occur is that in animals, we use these sort of brain atlases that have been averaged over a lot of animals. So we can say, if we want to go into the caudate in the rat, we would drill over this area and drop this many millimeters, and that's where we would leave it. In the human, for the implants, they do the brain scan first and then tailor the coordinates a little more tightly. There's just not enough money to do that in every rat. But after that point, it comes down to simple trigonometry. You have basically three measurements, and then you would lower your probe or needle. This approach also allows for experimental ablation. One of the most important research methods used in especially early behavioral neuroscience involves just destroying a region of the brain to see what happens. Now we do things a little more fine-tuned with optogenetics, but just even 20 years ago, this was the primary way to understand how something changed in the brain. If you think the amygdala is important, you go in and you blow it up. Really, you do it using an electrolytic lesion where you basically cause a lot of overexcitation and kill the area. But there are a variety of excited toxic toxins that can be used to create an ablation. I've also used electric current, like just a lot of electric current to basically you create an electrical hole in the brain. Like it looks like you've, basically fried a little segment of the brain. We can also perform recordings and stimulations of neural activity. Here I performed electrophysiology by putting basically a wire into the brain, and you can measure the firing of the individual neurons. Alternatively, I can apply electrical current similar to what I just described by performing an electrolytic lesion, but I can produce a little bit of electric current to cause the neurons to fire rather than to be overexcited and die. Recordings can be made chronically over an extended period of time. Like, so here, what I would have done in the past is I've, inserted as many as 80 different wires in the brain of an animal and then let them behave over the course of a month and just monitor how the neurons are behaving every day over that period of time. Alternatively, I have at times, more commonly, dropped an electrode in on the day of the recording and made a recording and then took my electrode out. So if we want to record the activity of a particular region of the brain, we would need to do this in the awake and behaving animal, which really requires the implantation of electrodes and surgery. We do this by attaching the electrode to miniature electrical sockets and bond these sockets to the animal's skull. There are many different ways and approaches that this has been done. Yeah, here is an example of what such an implant might look like. You tend to cover the implant with dental cement, which sticks to that bone, just like the bone in your mouth, very, very tightly. Even if there's a little bit of fluid, a little bit of water around, it won't degrade the integrity of the bond at all. And then after a period of time, you can just plug it in and hopefully get some recordings. Here's one example from the lab of Wolfram Schultz. What you see to the right here are what's known as peri-event histograms. The big top black histogram that you see, that is the aggregate of all the different trials of an animal behaving. Beneath that, you see one cell firing over and over and over again in about 40 different trials. And then the lines there, something happens. So in the first one up top, they give the monkey a juice reward, very sweet juice. And you see, in this case, dopamine neurons fire to that juice solution. Then they play a bell or something before the juice solution. And when that bell begins to predict the delivery of the juice, the neurons begin to fire at the juice. And then if you play the bell and then withhold the juice, the neurons shut down when the juice should have been played. This was some of the first data to suggest that dopamine neurons are involved in reward processing. The detection of these single neurons can be very useful and be harnessed in terms of medicine. This is some of the work that was first created by the lab of Miguel Nicolelis. Nicolelis started doing this with rats in an operant box, but he would record neurons from M1 of the motor cortex, measure their firing pattern, and then get to the point that he could create an algorithm and then play it back so that the animal could control motors and machines with their brain. So in this monkey, his left arm is tied behind his back, but he still has to, and his right arm is constrained. So the only way that he can feed himself is by reaching with this robotic left arm that's controlled by neurons in his right primary motor cortex. The same sort of technology has been applied to humans. Here is a soldier, who has been injured. Here, I'll be quiet and just listen.
Today, I'm really excited to share with you new results on how we are working to directly interface machines with the human brain. A DARPA-funded research team led by the University of Pittsburgh has demonstrated for the first time ever in humans the experience of the sensation of touch through a robotic prosthetic arm connected directly to the brain. The volunteer, whose name is Nathan, underwent a surgery to have 2 microelectrode arrays placed in his sensory cortex. This is a region of the brain responsible for identifying tactile sensations such as pressure. Those electrodes were then connected by wires to a robotic hand and arm fitted with tactile sensors in its fingers.
Index. Brain.
What you're seeing as the trials take place is that as the researcher applies light pressure to the robotic fingers, those physical sensations are converted into electrical signals that are fed directly back into Nathan's brain. Through this brain-machine interface, electrical signals are delivered as precise stimulation that his brain interprets as though his own fingers are being touched.
Index. Index. Ring.
Despite being blindfolded, Nathan can identify with nearly 100% accuracy
Pinky.
which fingers on the robotic hand are being touched.
20 out of 20.
What does this mean for the future of neurotechnology? DARPA has previously shown that a brain interface can be used to direct the movements of a robotic arm. Now, with this new development of adding sensation by directly sending signals from the robotic hand back into the brain, We have closed the loop between human and machine. At DARPA, we are always pushing the boundaries on what is possible.
Yeah, I've also seen it with a computer mouse too. So you know when right, if I were rendered paraplegic or quadriplegic and could not use my arms, I would consider being given the use of a computer mouse to open an incredible amount of enriching doors. It might be really the biggest thing that anybody could give me in that state. I would think. I have heard Miguel Nicolelis, the guy that sort of started all this, pour some cold water on some of these human studies that everybody's very gung ho about these days. While the technology is basically almost there, he is very concerned about the ethical side of this, and everybody's sort of wanting to jump the gun, in part, arguably, for their own ego. But then also, when you start getting very emotional about the one patient, like you have this one patient that you really want to give this treatment to, you can do much more harm than good at the end of the day, because you lose objectivity of the entire scientific advancement. Maybe the technology isn't quite ready to be rolled out to the individual quite yet, which is very frustrating. You don't want to be the person trying to tell this to the family members or the doctor that is eager to give this technology to their patient. However, if it goes wrong in one patient, the entire field can be set back forever. Maybe for 20 years, 50 years, or just that forever. Like, if there's one bad case, this is the sort of thing that could never recover. So you have to be very cautious when you approach these things, which is a very difficult thing to do. We also can measure the neurochemicals rather than the changes in the neural firing. This is mainly done using microdialysis. Microdialysis is a process in which substances are separated by means of an artificial membrane that is permeable to some molecules but not others. So a microdialysis probe consists of a small metal tube that introduces a solution into a section of dialysis tubing, a piece of artificial membrane that's shaped into the form of a cylinder sealed at the bottom. Let me think of so simply put, I've done this before. So think of the microdialysis tube as a cheesecloth, or, a sponge, if you will, like just a little tube with some holes, a little filter. So what happens is you put that in, and then you start infusing a fake brain fluid into the brain through this tubing, so that when that fluid gets down to where the brain fluid is, it would allow whatever chemicals are in that brain fluid to diffuse into the fake brain fluid, and that fake brain fluid to be pumped up so you can then measure chemically what and how much of different brain chemicals are in that region. So it's very powerful. One downside is it's relatively slow versus some of the other techniques. For example, when I've done microdialysis, I had to do these samples in 20-minute epochs. So in terms of behavior, that resolution is quite poor. A lot of things are going to happen in a 20-minute period. So how can you really say that dopamine is doing this during this specific behavior? It becomes difficult. So the technique that I've used more recently in my approaches is called in vivo voltammetry. This measures a neurotransmitter with a stereotaxically implanted microelectrode. Here you can measure things in 110 millisecond increments rather than 20 minutes. So you have almost near time resolution, but you're limited to measuring oxidizable chemicals. So while with microdialysis, I can measure glutamate, GABA, and dopamine, with voltammetry of those three, I could only measure dopamine. Here is a recording I guess that's not going to work, unfortunately. I will try to give you a, well, here, let me see if I can pull that up real quick in a different recording. I know that I just brought that over. Ah, there we are. So here is a voltammetry recording that I did in an animal. So in this case, the animal is responding for brain stimulation reward. Oh no, sorry. That was an optogenetics stimulation. The animal wasn't behaving there. But we will come back to that one. I'll show you that one in a minute. Here is a video of a rat, in this case an albino rat, rather than a Long Evans rat that you saw earlier. But here I'm measuring dopamine release as the animal is responding on a lever to receive electrical currents delivering directly to the dopamine neuron. So the animal is responding for dopamine neuron stimulation. When the Q light above the lever goes on, the lever is sent out. So the red there actually is, corresponded to the dopamine release evoked by the Q light. And then the second big peak there corresponded to the animal receiving the actual electrical currents. Wrong one. Right. All right. So that brings me back. Nope. Still the wrong one. Still the wrong one. Sorry about that. Now we are back to optogenetics. So, optogenetics would involve stimulating a particular brain region with electricity or an excitatory chemical that affects all of these neurons, and the result is unlikely to resemble normal brain activity, which involves the coordinated activation and inhibition of many uh, different neurons. So ideally, we would like to be able to stimulate or inhibit select populations of neurons rather than just depolarizing everything with electricity. So one way that we can do that is using optogenetics. Optogenetics allow us to do just that. They involve photosensitive proteins that have evolved in many organisms, even single-celled organisms such as algae and bacteria. This is what you see when you see the glowing organisms on the ocean lighting up in the moonlight. But researchers have discovered that when blue light strikes one of these proteins called channel rhodopsin 2, the channel opens and the rush of positively charged sodium and calcium ions then depolarizes the membrane, causing excitation. The use of genetically modified virus to insert light-sensitive ion channels into the membrane of a particular neuron in the brain can then either depolarize or hyperpolarize the neuron when light activates the appropriate wavelength. So what you can see here at the top are two graphics, one of channel rhodopsin being illuminated by blue light. So in this case, what I would do is I would put a blue laser right above the brain region that I infused a virus carrying the DNA to lead to the expression of this channel rhodopsin 2 ion channel. So you would infuse the virus and go ahead and implant the tip of the laser, or basically a tube, a tunnel that will lawfully channel the laser beam into the animal's brain and leave it so that it's located right above where the virus was infused. You have to wait about 1 month for the DNA that you put there to be activated and translate the ion channel that is then inserted into the membrane. Channel rhodopsin 2 would only respond to the blue light and lead to the influx of excitatory sodium ions. Halorhodopsin is what you see abbreviated up there, NPHR. Halorhodopsin is only responsive to yellow light and would lead to the influx of chloride ions, which would lead to a hyperpolarization of the neuron. So this would allow you to either turn on or off the neuron with blue or yellow light, respectively. And these effects are short-lived. You turn it on, you can sort of turn on the neurons like a light switch, on and off, to see how that affects behavior. I have and others have argued that this gets at causality. It certainly heads in that direction. Whether or not it's perfectly causal, not exactly so concrete. But using optogenetics, you can, for example, stimulate dopamine neurons in addition to other neural populations. Here is that would be actually here is an example of me performing a simple stimulation. So here, the animal, I'm doing both optogenetics and voltammetry simultaneously. There you could see the laser that I pulsed up above, and then that would then trigger the dopamine neurons, and then you're measuring the release of the dopamine neurons. So again, you can see the blinking, the flashing of the laser up above. So I applied it at 10 pulse, 20 hertz, manner of firing that's supposed to be reminiscent of natural burst firing of dopamine neurons. And then you can see the actual dopamine release that would occur and be measured at the terminal fields. So, I mean, if you're going back, I'm actually stimulating the dopamine neurons in the ventral tegmental area and measuring the release in the ventral striatum. So that is all I have for the first part of our methods presentation. In the second part, we will spend a lot more time on methods used in clinical populations like fMRI and PET imaging.